Now let's move from curiosity to clarity. This slide shows what genetic engineering really is. Genetic engineering is a precise process of altering organism genetic material by removing, changing, or inserting individual genes. As in the previous videos, we have seen that the genetics circuit is composed of three parts: promoter, coding sequence, and terminator. Promoter is actually the sequence which actually turns on the transcription and gene expression. Coding sequence is our gene GOI. GOI mean gene of interest. And GMO means genetically modified organism, because once we have moved our gene of interest into bacteria, it will not be a normal bacteria now. It will be called as a GMO, genetically modified organism. Okay, let's check in detail how actually the process of genetic engineering occurs. For this, we need three parts, we know, in fact, four parts. The part one is a promoter. Second part is a CDS, coding sequence. Third part is a terminator. And actually, we also need a fourth part, which is backbone. These are actually the extra chromosomal rings from bacteria. We have to actually cut them out open from two sides, one from here, one from here, with the help of different restriction enzymes. Then we have to put our these three parts into this cut open backbone. For example, here will be the promoter. Second part will be the CDS. The third part will be the terminator. Lastly, we have to do one more step, that we have to transfer this whole piece of DNA, including these three parts and plus one, the backbone, right? This blue part, into the bacteria. And this process is called bacterial transformation. Suppose I have to, let me write here, bacterial transformation. Some red part here, CDS part here, terminator here. Bacteria is not a normal bacteria now. It is a genetically modified bacteria, GMO. Okay, I hope it is clear to you people now. Let's move to another slide. Next slide. Yes, the process of genetic engineering, I have already explained to you all these steps. First, we have to isolate the gene of interest from humans. You can download the sequence from different websites, and then you can, once you have the sequence, then you have to cut open the backbone. This is the black backbone that I told you, that these are the extra chromosomal rings and bacteria. Usually they are circular like this. You have to use different restriction enzyme, as I explained to you before. uh, the restriction enzymes will cut them open from two ends, then they will be like this. Then you have to move your, the segmented part here, the yellow part actually is the same part that I have explained to you. It has contained three parts actually: the promoter, the CDS, and the terminator, right? Once, then you have to do the ligation. Ligation is a process in which these two ends will be sealed again, right? So it will be uh, into the circular form again. The last part is the transformation, bacterial transformation. This one I have also explained to you already. So once the bacteria have this whole chromosomal ring having your gene of interest with the whole cassette along with it, it will be called as a GMO, genetically modified organism. Okay, let's move to another slide. Okay, effective for genetic engineering. There are some microbes, they are considered very effective for genetic engineering and use in is in bioreactors. The first one is the bacteria. They are highly recommended and they are highly used because bacteria takes only 8 minutes to divide. So these are considered highly efficient for higher yields, higher production of your gene of interest. Second ones are the budding yeast, and the third ones are fission yeast. Budding yeast usually divides through a bud, and fission yeast actually divides in result of a fission. So these are the microorganisms that are effectively used in bioreactors. Next, we will see what are bioreactors. So this video will be a bit long if I explain in this video. So I'm going to close it here, and kindly keep watching and subscribe to my channel and like my videos. Bye bye.